My name is Christina Goethel. I am from Galt, California, which is just outside of Sacramento, and I am here at the University of Maryland Center for Environmental Sciences Chesapeake Biological Lab as a master's student under the area of specialization of environmental sciences. My project is looking at the effects of steadily rising carbon dioxide in the atmosphere and how it interacts at the air-sea interface, so it enters the water, and what that extra CO2 does to the water chemistry. So essentially what it does is it decreases the pH. So ultimately I'm interested in looking at how that affects Arctic animals, specifically a small clam called Macoma calcarea. My project is important because the Arctic is actually a great place to study stuff and then compare it to the rest of the world. So specifically with ocean acidification, because the water is cold, CO2 is more soluble. So we're expecting to see lower pHs in the Arctic first before regions like the tropics and other temperate zones, the ones in between the Arctic and the tropics. So it's a great sort of learning environment, and the Arctic also acts as sort of a global climate moderator. My setup in the cold room to study the ocean acidification effects, I have 6 tanks with 6 clams in them each. 3 tanks have water with pH 8.1, 8.2, which is normal seawater, and 3 tanks have pH of 7.8. And then I have 2 header tanks, which is where I control the pH, so it's just seawater, and one is just seawater, which is, magically already 8.2, 8.1. And then the other one, I have a system in which I can bubble a set amount of carbon dioxide into, and probes that measure that to make sure that it's at the correct 7.8 pH. And every day I go in and I measure the pH with a handheld probe in each of the 6 tanks. And I do a water change every 2 to 3 days, depending on what the pH is, to make sure that the 3 that are supposed to be at 8.1 are at 8.1, and to make sure the 3 that are at 7.8 are actually at 7.8. And I also measure temperature and salinity just to make sure that the other parameters of the water are proper for the clam's natural environment. Once the clams have been exposed to the 2 pH waters, this is a pilot project, so we're gonna use a probe to measure metabolic rate by measuring oxygen consumption, and then compare them to see if there's any differences, if the clams essentially have to work harder in an acidified environment.